Good afternoon, everyone. We're going to start this very exciting session on COVID-19. First, we're going to have our keynote speaker, Sunney Xie from Peking University. Sunney is the head of the BioPIC and the ICG. And in the past half a year or so, he's been doing amazing work using single-cell technology in producing neutralizing antibodies against the COVID-19. Very impressive work recently published. We're very excited that Sunney will be presenting this part of the work. Welcome, Sunney.
Wow, thank you, Zemin, for the kind introduction. I also would like to thank Guoji for giving me the honor to speak first. The COVID-19 pandemic has proven catastrophic for public health and the world economy. It has been an emotional and stressful time as China and the world was gripped by the spreading virus. As scientists, we all try to make a difference. I'd like to share with you what we have learned and accomplished during this past months. You all know many vaccines are now under clinical trials. Before safe and effective vaccines are widely available, there's a high demand for effective drugs against COVID-19. So here's a list of drugs that have been tried to treat COVID-19. If you could see the last column of summary, the conclusion is that few of them are effective. We certainly do not have an effective cure at this point. So what are the standards for effective drug? It would significantly cut down the proportion of patients with mild to moderate symptoms turning into severe. Second, significantly reduce the damage of patients' lungs and other tissues. And third, quickly cure mild cases, thus shortening the hospital stay. Now, you all know that plasma therapy has proven effective, yet it couldn't be largely applied due to limited supply. One patient needs 1,200 ml of blood from from 3 or 4 recovered patients, or convalescent plasma. The effective component of convalescent plasma is the neutralizing antibodies generated by the human immune system. What's neutralization? When an acid comes, a base can be neutralized, can be used to neutralize the acid. When a virus comes, neutralizing antibodies can prevent it from infecting cells. Compared with small molecule drugs, antibody drugs have few side effects. It's very specific. So our goal is to identify the neutralizing antibodies from convalescent patients, make them in vitro through mass production, and then inject them into patients. This is the Y-shaped antibody with the heavy and light chains. SARS-CoV-2 is an RNA virus. This S protein on the surface of the virus is a trimer that has a binding domain, RBD, the red part. Chinese scientists determined the genome sequence of SARS-CoV-2 in less than 2 weeks after the outbreak, then shared it with the whole world, which provided the basis for diagnosis and therapy. Later on, a few groups quickly reported the structure of the S protein and the binding to the ACE receptor, which is on the membrane of human cells, such as lung cells. So neutralizing antibodies bind to the S protein, which would prevent the virus from binding to the human ACE receptor. Here are 3 successful attempts of neutralizing antibodies to treat human diseases. In particular, the US FDA approved the treatment of multi-drug resistant HIV in combination with other antiviral drugs. In the case of Ebola and MERS, effective antibodies, neutralizing antibodies, were also developed, but the epidemics ended before clinical trials got finished. So I particularly want to mention the work of Michelle Nussenswick at Rockefeller. He first looked at the neutralizing antibodies of blood from different HIV patients using single-cell clonal amplification. Now you see these 4 different patients have different types of antibodies indicated by each color. Now the green ones are not potent ones. The other colors indicate potent antibodies. The number 4 has more type of potent antibodies. So this really highlights the heterogeneity among different individuals and the need for single-cell genomics. Suppose an HIV patient whose blood has high neutralizing potency. How would you know if he or she has a large number of mediocre antibodies or a small number of highly effective ones? So we must determine the distribution. This is obvious for people working in our community, of course. However, the conventional way of single-cell clonal amplification was just too time-consuming. Now, in recent years, many groups have been pushing for high-throughput single B-cell sequencing. So these are the 4 papers in the past 4 years. My own group were not actually doing this at all before the pandemic. The technology was ready, and we put it into medical use for human neutralizing antibody with very limited time under the pressure. So it worked, and it worked very well, of course, through hard work. Our efforts began on the 3rd day after the Chinese New Year. Richard Cao, the only Harvard group member who joined my PKU group, he and I returned to Beijing on the same day. So at the beginning, we work on coronavirus diagnosis. But when we realized that our expertise, single-cell genomics, could really accelerate the search for the effective neutralizing antibodies against COVID-19, we were thrilled. We started reading literature, designing experiments, borrowing reagents and equipment. Everybody was extremely helpful. On February 2nd, we went to Beijing Yuan Hospital, a designated hospital of COVID-19. We met President Jing, who immediately agreed to collaborate by providing blood samples from recovered patients. Bing and Xianghua, 2 researchers of Yuan Hospital, were trained in our lab by Richard and Cheng Yang, who is our sequencing facility manager. Then they 2 returned to their hospital's P3 lab wearing this heavy protective gears. They were supposed to work no more than 5 hours per day, but they always overworked. In the end, they were isolated for almost 3 months. at Peking University, Richard and Cheng Yang have been fighting day and night to collect and analyze sequencing data. This is the cell typing for the convalescent patient's blood by single-cell RNA-seq. You can see different cell type, different types of B cells. So they develop at the beginning from the bone marrow, then grow in the blood. And we were most interested in the memory B cell and the plasma cell. So at the beginning, each B cell undergoes VDJ recombination. As most of you probably know, the work won the Nobel Prize in 1978 by Japanese scientist Tonigawa. The chromosome 14 of the human genome has 40 different sequences of V sequences and 23 different sequences of D and 6 different sequences of J type, like 3 sets of cards. The recombination of VDJ is like drawing one card from each set of VDJ cards, which has the diversity of 40 multiplied by 23 by 6. That's 5,520 in the heavy chain encoded by chromosome 14. So similarly, chromosome 2 has V and J sets, but not D. So that encodes the light chain, which adds more to the diversity of total of 10 to the 7th to 10 to the 8th types of sequences in a human body. Now, in addition to VDJ recombination, point mutations provide more types of sequences. And each sequence results in a different protein with certain molecular shape and interactions, hence varied binding affinities for a specific antigen. Now, what's interesting is that this 10 to the 7th to 10 to the 8th types of cells, each cell has a unique sequence, they exist before an antigen enters the human body. On the membrane of the B cell is the B cell receptor, BCR, similar to the Y-shaped antibody, except they're just bound on the membrane surface. So when antigens such as SARS-CoV-2 comes, binds to the BCR, recognized by the BCR, then what happens is the B cell gets activated and start to divide. Otherwise, they wouldn't divide. And you start to proliferate. And 1 becomes 2, 2 to 4. And then as a consequence, some of them become the plasma cell, which secretes antibodies into the blood. And that they become the neutralizing antibody against the virus. And the rest of them become memory B cells, something we're more interested in, because on the surface of memory B cell are these BCRs. We want to isolate them, the memory B cells, from the blood with their BCRs. So at the beginning, we only used the 10x high throughput VDJ sequencing kit with no functional selection. We sequenced large number of cells which are binned, from 10 recovered patients in order to feed the high throughput sequencer. Unlike Michel's work, we were not looking for Superman. We were looking for super antibodies. The high throughput of the next generation sequencing can do 10 patients together, 10 to the 5th cells at the same time, as you all know. you see these cells, six cells, have almost identical have exactly identical VDJ sequences, which means they all came from the same parent cell. That is to say, they are enriched and amplified as a result of this activation that I just mentioned. So our assumption was that the higher the enrichment, the more useful this particular sequence is to the immune system, as it is selected by the complicated evolution system of the immune system. So once we determine the sequence, we of course could use it to synthesize the protein in vitro. We actually asked a company to do it. In doing that, we get the recombinant protein and then test their binding affinity and neutralizing potency. Believe it or not, even for the first patient, we were able to find an enriched sequence that resulted in an antibody with neutralizing ability. However, it only accounts for 1%. So if we have 100 sequences, one of them had the neutralizing potency. So we wanted to improve the yield. So what we did was to use this functional selection, attaching the S protein to this magnetic bead and use a magnet to pull this memory B cells out. And that gave us a yield of successful sequences up to 20%. And then we use RBD linked to the magnetic beads, and that further improved the efficiency. So this was our first Cell paper. We reported 58 sequences, along which we found 14, now 20 potent neutralizing antibodies as candidates. And I want to stress here, compared to the other groups at the early stage, the number of cells we went through was at a much, at least one order of magnitude. And the high throughput technology really gave us the ability to select better neutralizing antibodies, because this 10 to the 7, 10 to the 8 type are there, and we only get to sequence a small fraction, say 10 to the 5th. So it's really a probability matter. The more we sequence, the higher the throughput, the large chance we have to find a good one. So this is the IC50 curve. That's the neutralizing curve. The horizontal axis is the antibody concentration, and the y-axis shows the percentage of cells that get neutralized. So IC50 is the concentration at which the inhibition reaches 50%, which is 7 picomolar for pseudovirus, 100 picomolar for authentic virus. Similarly, IC80 is at 80%. So IC50 or IC80 indicate the drug dosage. The lower they are, the more effective the antibodies are, the less the dosage. Some of you might know that Trump received 8 grams of antibody made by Regeneron. That is a very high number compared to the normal dosage in cancer therapy. PD-1, PD-L1 is usually under 1 gram per individual. So if there were a more potent neutralizing antibody, patients would receive a lower dosage. Xiaodong Su and Junyu Xiao, 2 structural biologists at Peking University, determined the structure of neutralizing antibodies bound to the S protein using cryo-EM. So the blue bottom is the trimer, the red one is the RBD, and the purple one is the antibody. It is evident that the antibodies binding block S protein binding to the ACE2 receptor. We found most of the potent neutralizing antibodies bind to RBD, not only from the cryo-EM structures, but also from the comparison between the RBD pooling and the S protein pooling down. Neutralizing antibodies have two usages, one stone that kills two birds. First, it is a therapeutic drug. Second, the prophylactic use as a temporary prevention for people like patients, family members, or medical staff. The prophylactic period, as we measured, is around 3 weeks, but can be prolonged to 3 months with a modification of the antibody protein. Dr. Qin Chuan's team in China first established the mouse model for humanized ACE2 receptor. So our animal tests were completed in her lab. They infected the mice with SARS-CoV-2. Then after 2 hours, gave our most potent neutralizing antibody 386-2 with a dose of 20 milligram per kilogram. The green one is the therapeutic experiment. The blue one is the negative control for which irrelevant antibody was given. So after 5 days, a high viral load was detected in the lung. This is for the control in blue color. And the viral load was reduced by a factor of 2,500 in the therapeutic experiment. This is a significant drop indicating the effectiveness of the antibody at this particular dosage of 20 milligram per kilogram. If the prophylactic experiment, in this experiment, we gave the 386-2 1 day before the infection, then after 5 days, we detect no virus in the mouse's lung. Now, this shows the body weight change with time. You see both therapeutic and prophylactic group show little change. A slight decrease in weight could be due to the shot, whereas in the negative control, this decline of the body weight. So I'd like to share with you our results from our second cell paper, which is published online. Junyu and Xiaodong have more interesting structured data, and Qin Chuan's lab made the testing on hamsters, which really give us a much clearer sense of regarding the effectiveness of the drug. Then we developed a new cocktails therapy. So these graphs show the potency of the neutralizing antibody. 386-2 is order or 2 magnitude better than these other antibodies that we found. So what is the structural basis? In this cryo-EM structure, you see here's the S protein trimer, and there are 3 RBDs, 1 up and 2 down. And the structure shows that our 386, 368, I'm sorry, -2 binds to all three of them, either up or down. Okay, so this means the stoichiometry of our antibody binding to the S protein is different from the rest of them, especially the one I showed you with the previous structure. Yeah, so this explains from the structural point why our 368-2 is most potent. Now, Qintran's team used a new hamster model, which is more advantageous than the mouse model because the hamster is heavier and shows more severe symptoms than the mouse. It can actually, the hamster can die of COVID-19, whereas the mouse only had the light symptom. So under the previous dosage of 20 milligram per kilogram, we look at the effect of the window of dosage extending from 2 hours to 48 hours with almost no change relative to the negative control. You see the metabolism of a mouse or a hamster is much faster than that of a human, so everything is accelerated. So roughly multiplying by 7, then you can get the number for humans. So for human, 48 hours is about 2 weeks. And that corresponds to the time developed into moderate or severe symptom. So the antibody couldn't be suitable for severe cases, as the severe case should not be categorized. It should be categorized to another kind of disease, and that is the immune system were greatly compromised. So the body weight change were confirmed by the pathological results, which are certainly more reliable. We then evaluated the dosage dependence, changing the dosage from 20 milligram per kilogram to 10, 5, and 2 milligram per kilogram. Now, of course, the lower the better, right? Even at 2 milligram or 5 milligram per kilogram, the antibody drug was still effective compared with the negative control. And this was also confirmed by the pathological results. So finally, I want to talk about the pairing of antibodies of non-overlapping epitopes. So this is 368-2 on RBD, and this is the antibody 629 on the RBD. Now, these two epitopes do not overlap, so this is the crystal structure of both antibodies on the same RBD. So the idea was, if the virus undergoes mutation, one antibody might become ineffective, but the chance for both of them undergoing mutation is extremely small. So therefore, the use of a pair could avoid mutation escape. And this was first demonstrated for the case of COVID-19 by Regeneron. Okay, Dr. Yuan Wang's group in China recently evaluated the neutralizing ability of our two antibodies on the reported coronavirus mutation strains. The most commonly observed mutation strain, 614G, luckily didn't have mutation in the RBD region, hence free of drug resistance. But these three strains, L F452R, V4H3A, and F490L actually did have mutation escape. Our 368-2 no longer have neutralizing effects. So luckily, these three strains have very low abundance in nature so far. Now, as expected, if we use a pair of non-overlapping epitopes, the neutralizing power remains the same, and this is the neutralizing curve of the combo. So finally, I want to show you our latest result, a very interesting one. So if we see how the IC50 or IC80 in this case change as a function of time. So we show the value at 2 time points. So you see for each antibody alone, this 593 is actually equivalent to 368-2. It's our naming system, okay? And the other one is 604. That's very similar to 629 I show you. Okay, so these are the IC80 on the 3rd day, and this is on the 5th day. You see, they are greatly reduced with time. Why? This is due to the mutation escape under the evolution pressure of the antibody drug. So if the drug kills the virus, then the virus gets more mutations, not in the region of the where the drug, the antibody binds. But what's so great is if we use this antibody pair, you see the IC80 does not change with time. Okay, so this is a very pleasant result. So the conclusion is the pairing is the way to go. So finally, as a disclosure, I mentioned the potential conflict of interest. My 2 students and I have formed a startup company called Syngolomics with this drug as the first product. Our goal is to use single-cell genomics to develop antibody drugs. And Syngolomics collaborated with Beijing, a big pharma, to conduct global clinical trials of these neutralizing antibodies. And here I have a list of clinical trials of COVID-19 neutralizing antibodies around the world. And Regeneron and Lilly are ahead of the game in terms of the trial dates. And I want to quickly summarize what I have said. High throughput single cell transcriptome and VDJ sequencing speeds up the development of clinical use neutralizing antibodies. We have found over 20 potent neutralizing antibodies from more than 400 recombinant proteins produced by analyzing 8,600 B cells VDJ sequences from about more than 60 convalescent patients. Cryo-EM structures show effective neutralizing antibodies bind to the RBD of spike protein of SARS-CoV-2. The most potent ones binds to all 3 RBD, 1 up and 2 down. A pair of neutralizing antibody with non-overlapping epitopes can effectively avoid mutation escape. Our animal test demonstrated that our potent neutralizing antibodies exhibit high therapeutic and prophylactic efficacy against SARS-CoV-2. I wouldn't be able to tell you this story if I haven't worked with this really talented, hardworking students and colleagues and collaborators. Many of them I got to know only after the pandemic. All these institutions listed here are involved in this work, and I want to thank the support from Beijing municipal government, Ministry of Education, Ministry of Science and Technology, and I want to thank you for your attention.
Thank you very much, Sunney, for the inspiring story. Now let's turn to Q&A session. So we have a few questions, and I guess I'm supposed to read off the question to you. So the first question is from Cheyenne, and the question is, if you sequence 1 million B cells from a convalescent patient, how many of them on average would have an IC50 below roughly like 50 nanomolar? So, what's the chance you get such a good antibody?
From one individual, wow, I gave this number 1 million. 10 to the fifth, we get 1, right? Okay, so 1 million will be about 10. But you see, that's only a small fraction, right? So I always get this question. All these people, they went out from the hospital, right? So they had effective antibodies. Why it was so hard to catch them? And that is because we are sequencing a very small fraction of them, right? We're not sequencing 10 to the 7th to 10 to the 8th. If we do, then we find all of them.
If you do 10x genomics, you're able to sequence about 10 to 20 cells per assay. So you're not at the scale yet.
Yeah, so we have, see, we've been, we could do it one person at a time, right? But like I mentioned, I mean, it's heterogeneous. So in the end, we decided we want to do 10 people at a time because we want to get the best one. We don't care the Superman. We only care about the super antibody. So I mean, roughly just like we're sequencing 1 part per thousand or 10 to minus 4. But so in that set, we got a few. I mean, that's roughly the number. Yeah.
Okay. The second question came from Callum Parr. The question is, do patients treated with exogenous antibodies or plasma go on to develop a robust endogenous immunity to COVID-19?
No, I don't think so. I mean, the neutralizing antibody, they just, if they're present in the blood, they stop the infection, and then they all have lifetime, they go away, and then the people will go back to normal. Now, in order to, this is not like a vaccine. Vaccine, on the other hand, would create the immune response that would have the memory effect that lasts hopefully forever. But that is really depends on the disease. HIV, nobody have found the vaccine, and so there was there were side effects of ADE, antibody-dependent enhancement. You could have mutation that makes the antibodies previously generated by vaccine become ineffective. And then they're there, they go there to find the virus, but they don't stop them. They're not strong enough to stop them, and then they got pulled into cells because there's FC receptors. So the ADE could be a problem if there are a lot of antibodies, but they don't do anything. They're not effective. So there was actually a theory, after the Spanish flu, and the first time, less than 1 million people died. It was sort of similar to COVID, but then the second year, 10 times people died, and that one theory was due to ADE, right? So this is if you were to have immunity among a large population. I mean, this could be a potential problem. But if you just take this neutralizing antibody as a preventative drug, this wouldn't happen. But it's more expensive than vaccine, right? So.
Yeah, I know we're running short of time. Just one final question. Maybe you can give a very short answer. The question is like this. So this from somebody. They said, I read from the news that some college student in the U.S. try to get virus and then sell their blood after recovery to cure other patients. Can you comment on such effort if there's no drug or vaccine available?
I don't know.
Okay, I know. It's just they purposely get sick and then they would, sell their blood.
Sounds
apparently, U.S. have this approach. No. But anyhow, this is probably kind of a hard question to answer.
I got to sounds pretty dangerous to me. Was this for COVID?
For COVID, yeah, especially young people who have a low mortality rate. Yeah. Anyhow, so this is more of an entertaining question. So yeah, thank you very much. Yeah. Okay.
Thank you.
Yeah, thank you very much, Sunney, for the really inspiring presentation.